Process of genetic engineering is not like sexual reproduction. Let's say you want to create a corn plant that produces its own toxic insecticide. So first you find a natural toxic insecticide like Bt from Bacillus thuringiensis in the soil bacteria world. You take the gene out that produces the toxin that breaks open the stomach of insects to kill them, and you make millions of copies of that gene. Then you put it into a gene gun, and you shoot that gun into a plate of millions of cells, and then you identify which one of those genes got into which of those cells, and then you clone those cells into a plant, and now every cell of those corn plants has a gene-sized spray bottle that can kill insects. Now, the process of genetic engineering, whether it's done by gene gun or bacterial infection, and followed by cloning, creates massive collateral damage in the DNA.
Quite a lot of difference. We were able to do some more elaborate statistics and actually hunt down some hypotheses within the statistics that we used. And the other thing that we did was that we know that pigs are not just fed a GM crop at a time. They're fed combinations of GM crops. They're fed GM soy and GM corn at the same time. And also, I mean, the main variety of GM soy that we have is Roundup Ready soy. So this is designed to be resistant to being sprayed with Roundup and still survive while the weeds around it die. But within the corn, and that's the main one within soy, you know, it's basically just Roundup Ready soy. But within corn, there are so many varieties of corn. Some of them are designed to be resistant to not just glyphosate, but other herbicides individually, and there are lots of different insecticidal proteins that are put into that the plant is genetically engineered to be able to produce. So there are a whole variety of different genetically engineered corn plants varieties out there. So therefore, if you're a person and you're eating genetically engineered corn, you're eating probably a variety of different GM corn varieties at the same time. And then if you're eating a diet in the United States, in a meal, you are probably eating GM soy and GM corn at the same time and different varieties of corn all at the same time. And there is no regulator in the world that requires genetically the GM industry to do feeding studies on animals where you're feeding more than one variety of plant, GM plant, at the same time to the animal. So and we know that within pharmaceutical drugs, that there's a thing called polypharmacy, that if you feed somebody, you know, an aspirin, for example, there might be some certain side effects. But in addition, if you feed that person some other pharmaceutical drug, the two drugs can interact to produce outcomes that you can't see by feeding just one drug or the other at one time. And so I was interested also in the effects of the equivalent of polypharmacy, if you like, when you eat more than one GM crop. And so with these pigs, we fed them what pigs usually eat in the United States, which is genetically modified soy, Roundup Ready soy, and we also had a couple of different GM corn varieties being fed to the animal at the same time. So we were in effect feeding 3 GM genes and their protein products to these pigs at the same time. So these pigs were eating the Roundup Ready gene and its protein product, and 2 Bt proteins, and the genes and the proteins from 2 Bt genes, which are designed to produce insecticidal proteins. And I suspect that the reason why we got such strong stomach inflammation was because of the interaction between the proteins that the animals were eating.
Pay close attention to what you are about to see. These images will be seen around the world. These rats have been fed with transgenic corn during their entire life cycle. The tumors they suffer from are enormous. The study that revealed the effect of the GMOs on the health of these rats has just been completed. It benefited from exceptional financial and technical means. We were able to witness the study in its entirety. It lasted for 2 years. Following years of doubt and controversies, will we finally know the truth about GMOs? Would we then be faced with a health crisis of global proportions?
This is the longest, most thorough study to have been carried out on a GMO, and we have also carried out a joint study on the most used pesticide in the world, the Roundup, which is a herbicide. It belongs to the main pesticide category. We spent over a year trying to obtain transgenic corn because no GMO manufacturers were willing to provide them. This is because Monsanto requires contracts to be signed stipulating that the seeds must not be used for testing and an arrangement must be made prior to the testing. We wanted our study to be independent.
This is the first time that a 2-year study has been conducted on GMOs. And there lies the big difference. Furthermore, we tried to evaluate a maximum of biochemical and biological parameters with repeated blood tests, weighing of the specimens, and urine tests. We studied the hepatic and urinary parameters, as well as the blood parameters, all the parameters that can be studied. We also studied the hormonal parameters, which has never been done in previous studies. This experiment is a world premiere.
Up until now, the corn NK603, the very one selected for the study, had only been tested over a 3-month period, and that in a test environment entirely controlled by Monsanto, its manufacturer. Furthermore, concerning the Roundup, also manufactured by Monsanto and which is used for growing corn, only its active agent, glyphosate, had been assessed. But one needs to understand that the manufacturers, in order to enable the active agent to penetrate the plant cell successfully, use additives that are sometimes more harmful than the actual glyphosate.
We had 2 clear objectives. Firstly, to conduct the studies exactly according to international requirements, but making sure to be very thorough when it comes to analyzing the results. Secondly, we want to determine if there are secondary effects. The only way to do this is to carry out tests on animals and then to examine all the blood parameters whilst paying close attention to their organs.
At this stage, it might prove useful to know exactly what GMOs are used for. It is a plant that has been developed either to produce its own insecticide or to be tolerant to a herbicide, thus enabling it to be exposed to massive doses of it without dying. One can understand how GMOs could be useful to farmers. For example, they would be able to spray their entire field with Roundup, killing all the weeds in the process and never harming the genetically modified plant. On the other hand, it is hard to see the benefits for the consumer, who will effectively be ingesting substances coming from a plant that has absorbed pesticides. That is why it is crucial to test not only the GMO, but also the GMO with its residue and the pesticide on its own.
This type of testing had never been done before because the corporations doing the tests would do them on GMOs with the Roundup, which meant that we had no idea what between the GMO and the Roundup caused the pathologies, or they did them on GMOs without specifying that this was the case. So what we decided to do was to address the question, are we dealing with the effects of the GMO or the pesticide? During the last 10 years of testing, the National Commission did not envisage a pesticide effect.
Therefore, Europe does not even demand that chronicle toxicity tests be systematically carried out on animals, and when they are required, it is the manufacturers who decide the protocol to be used. For example, the Amflora potato was only tested on a group of 30 rats, 10 of which were being fed the GMO. The Monsanto corn was tested on 400 rats. 80 of them were fed the transgenic plant, but only 40 of them were then subjected to blood tests. Either way, none of these studies lasted longer than 90 days.
What is represented by this figure is that certain groups of rats, male and female, have ingested GMO corn. GMO corn with Roundup residue and Roundup on its own. Every time we go up a level, a rat dies. The limit is 10 due to the number of rats that constitute a group. The dotted line represents the females of the group who die but have a completely normal diet. Only 2 animals die spontaneously after 550 days. On the other hand, every female that has been fed on the GMO with Roundup or Roundup diet has died more frequently than the other rats. Some will challenge the relevance of such results, but what is undeniable is that in the group that has been exposed to the GMOs, there is a 600% increase or 6 times more deaths compared to the control group. As far as medicine or biology is concerned, there is no need for further testing.
An important fact is that all the studies carried out on the rats are done over a 3-month period. It so happens that none of the effects are visible until the 4th month.
Well, initially we found that the larynx or the voice box in exposed males didn't grow properly. And this was an indication that the male hormone testosterone was not being produced at appropriate levels. And eventually we found that not only were these males demasculinized or chemically castrated, but they also were starting to develop ovaries or starting to develop eggs. And eventually we discovered that these males didn't breed properly, that some of the males actually completely turned into females. So we had genetic males that were laying eggs and reproducing as females. And now we're starting to show that some of these males actually show, I guess, what we'd call homosexual behavior. They actually prefer to mate with other males.
A single independent study conducted on the impact of genetically modified food shows that it damages organs, it causes infertility, it causes immune system failure, it causes holes in the GI tract, it causes multiple organ system failure, which causes both men and women who eat it in the form of any product to produce antibodies to sperm. If the men eat the epicite gene, they produce antibodies to their own sperm, rendering them irreversibly sterile. If women eat the epicite gene, when they have intercourse, their bodies produce antibodies to the sperm that has been deposited, and they become infertile through the destruction of the sperm. Now, DuPont and Monsanto formed a joint venture, purchased the epicite firm, and, quote, commercialized the epicite gene. Do you want to know if the food that you're eating contains the epicite gene? Sure you do. How about the food that your children are eating, or your grandchildren? Sure you do.
The very idea of GMOs is a death knell for biodiversity. Biodiversity means the integrity of life. GMOs mean appropriating life to own it and patent it. And because there are patents related to GMOs, and the only reasons GMOs are being introduced is to own seeds and collect rents and royalties, corporations push the handful of crops that they have genetically modified. So you can see in the last few years, cropping systems have been reduced to corn and soya and cotton and canola, when we have cultivated and eaten thousands of crop varieties and species. The uniformity and monocultures that are being pushed by GMOs are driving out diversity directly, but they are driving out diversity because instead of using less more efficiently, which they always claim, that land we are using more efficiently, they're having to invade the Amazon, they're having to invade the grasslands of Argentina for a very expansive and wasteful agriculture. And when you spray Roundup on Roundup-ready crops, you are literally killing food and diversity.
Is quite unique because it's not only based on literature, scientific literature, but it's also based on the 110 national reports that we have received so far by government, just indicating the status of biodiversity at local, national, and global level.
However, the report makes for grim reading. It reveals how the Earth's ecosystems and species are buckling under the strain of unsustainable development. Based on data supplied both by the scientific community as well as directly from national governments, The report reveals that the target agreed by world governments at the 2002 World Summit on Sustainable Development to achieve by 2010 a significant reduction in the current rate of biodiversity loss has not been met. Indeed, not a single government submitting national reports claims that their 2010 biodiversity target has been completely met at the national level. In synthesizing this evidence, the report states that the consequences of current trends are much worse than previously thought, highlighting the impact on the poor in particular, who stand to suffer disproportionately from potentially catastrophic changes to ecosystems in coming decades.